for my dead body. Get there too. Okay, yeah, so I'm going to put a pin in that conversation because I want to get through, but I just want you to think that we used to use bacteriophage therapy for treating people, and then antibiotics came along. We don't really use it anymore. We still kind of use it for random stuff. Anyone in here like lunch meat? They spray it with bacteriophages. So if you look under a lot of your lunch meats, it will say treated through viral phage therapy, but they don't advertise it because a lot of people are like, I don't want viruses on my food. But then you're the smart microbiologist, like, no, it's bacteriophage, it doesn't affect human cells. They're just using it to kill listeria. Okay, let's go to eukaryotes, and we'll get through this pretty quick. Differences to note. How they get into the cell. How they biosynthesize, and this has to do with, eukaryotes having a nucleus and prokaryotes not having a nucleus. And how they release, okay, because getting in and getting out are kind of reflective. Okay. So how they get in. A lot of cases for eukaryotes, there's two ways they get in, and this is either A, membrane receptor endocytosis. We went over it way earlier, I can't remember the exact name. Well, anyways, it's where the cell attaches to the outside. It's very specific lock and key, then it's taken in. Receptor-mediated endocytosis, that's correct. Yeah, there we go. The other way is endomembrane fusion. In this case, you can think of a virus, and it has to be enveloped in this case. A virus has an envelope. That phospholipid envelope is going to match the lipid membrane of the host, and they're going to fuse together and become one cell. Right? And actually, the host is way bigger, and you can think of this as a tiny bubble just kind of joining into the bigger bubble. It dumps its internal content. Eukaryotic viruses are very specific, and especially within something like a mammal where we have differentiated tissues. Some viruses will only affect certain types of tissues. Example here, polio is only going to affect stuff like nervous tissue versus flu is only going to affect respiratory tissue. This is known as tissue tropism. And sometimes they can change their tissue tropism within different stages of their infectivity. And we'll discuss a little bit about this when we talk about latent viruses like chickenpox. All right, so 6 steps here. Attachment penetration, so receptor-mediated endocytosis. In this case, this virus does not have an envelope. It attaches on, and then when it bubbles inside, it takes part of the host membrane with it. So it's in this vesicle. Because it's within this vesicle, we have an extra step here. Instead of going directly to biosynthesis, it's going to have to uncoat itself first. So don't forget uncoating. Once it gets in there, it has to take off its jacket. It's inside the house. Makes all of its stuff. Okay, if it's a DNA virus, it's going to do this within the nucleus. If it's an RNA virus, it'll do this within the cytoplasm. You don't need to know that specifically. It's going to assemble it together. Great, this is analogous to maturation. Then release. And in this case, release. Notice it's release and not lysis. Why do we have release instead of lysis more commonly with eukaryotic viruses? So it doesn't destroy the membrane. Let's elaborate more. Why don't you want to destroy the membrane?
Because it'll set off the system.
Keeps the cell alive. Why do you want to keep the cell alive? You don't want to kill the cash cow. Right? If you kill the host too quickly, then it's not going to have time to spread. It's going to do this to a degree so that it can spread from host to host. That's a form of viral eukaryotic evolution. What's the central dogma of biology? Y'all recall? We start with genetic material. We start with DNA. What's the process of turning DNA into, what's that called? Yeah, RNA. DNA to RNA, what's that called? Transcription. Okay, we have RNA. RNA goes to? RNA is used by ribosomes to make, yes, proteins. RNA to protein, what's that called? Translation. Okay, that's what we call the central dogma of biology. So DNA viruses are going to follow that central dogma, but because, as I mentioned, we have double-stranded DNA, single-stranded DNA, double-stranded RNA, single-stranded RNA, positive single-stranded RNA, negative single-stranded RNA, that central dogma isn't necessarily always followed. Right? So double-stranded DNA is going to follow that normal flow. Transcription to RNA, translation to a protein. Single-stranded DNA has to first make itself double. So what single-stranded DNA does is it'll form a complementary strand for itself, what they call cDNA, and then it goes through the normal central dogma. When we get to our RNA, it's a bit different. So if we have double-stranded RNA, it's composed of both positive single-strand and negative single-strand. If you have just positive single-stranded RNA, it skips most of the steps of our central dogma. It just translates to protein. Okay, the positive single strand acts like messenger RNA, and it goes directly to a protein. Very quick, but it does. But if it's negative single strand, it has to go through some extra steps, and this is where the most complex part gets of this. We have negative single-stranded RNA. It has to bring along a specific enzyme known as viral RNA-dependent RNA polymerase, or RDRP. It has to have that because what RDRP does is convert it to positive, right? So it'll take a negative single-stranded RNA, this enzyme converts it to a positive single-stranded RNA, so then it can go and it can make proteins. Question?
Do you want us to know all of these life cycles?
You need to know that there are double-stranded, single-stranded DNA, double-stranded, single-stranded RNA, negative and positive sense, okay? That's ways that we categorize viruses, because in order to treat a lot of viral infections, we have to know what their genetic material is, and that's going to come important in chapter 14 when we talk about antiviral drugs. Bless you. The most important part that you need to know, because there's going to be a question over it, what enzyme does negative single-stranded RNA need to bring with it in order to make proteins, in order to translate? Viral RNA-dependent RNA polymerase. And you can bet you that I change these up and say viral DNA-dependent RNA polymerase, viral DNA-dependent RNA polymerase, host-dependent, okay, RDRP, right? RNA-dependent RNA polymerase. We good? I'm giving you all kinds of answers today. Yay. It is of note that you don't necessarily need to know for this class, but if you are a double-stranded RNA virus, they also bring this enzyme along too, and so they can get more bang for their buck. Because if you're a double-stranded RNA virus, you split into your positive and negative sense strands. It'll use its positive sense strands to translate, but it says, well, I don't want to waste my negative sense strands, so I'm going to take that enzyme along so that I can convert it to positive. All right. Sometimes we go backwards through the central dogma. And that's in the case of some single-stranded RNA strands, like HIV. Right? Positive single-stranded RNA sometimes doesn't want to go just directly to make proteins. Actually, it wants to go become DNA. And so it brings a specialized enzyme with it. So if a RNA virus wants to go to DNA, it has to bring along reverse transcriptase. So we talked about the name of transcription, going from DNA to RNA, just like the name suggests. Reverse transcription reverses that. You go from RNA to DNA. So what they'll do is they'll convert their RNA to complementary or single-stranded DNA. Then they'll make a reflective template into double-stranded DNA. And after they make that double-stranded DNA, then they integrate themselves into the chromosome. So they go backwards from RNA to DNA, and they make that DNA go into the host, goes into us, goes to animals. That integrated DNA is now known as a provirus. What's the integrated DNA called in a bacteriophage?
Prophage.
Prophage. What's the integrated DNA called in a eukaryotic virus, retrovirus?
Provirus.
Provirus. What's the integrated DNA called in a bacteriophage?
Prophage.
What's the integrated DNA called in a retrovirus? You notice how I'm saying this like 3 times? There's like 2 or 3 questions on the exam for that, and I say this every time in the class, and then people come and they miss all those questions. Know the difference between a provirus and a prophage.
Could you say it one more time?
Prophage, integrated DNA in a bacteriophage, so a lysogen. Bacteriophage is associated with prophage. Their names match. Retrovirus will form a provirus. So eukaryotic retrovirus, provirus. Both have pro, right? Just associate the phage with it. So that's how HIV works. You don't need to know this specifically, all these steps or anything like that. This is demonstration purposes only. So we have positive single-strand HIV. There we go, it's enveloped. Endomembrane fusion, see what that's showing you? Remember how I told you the little bubble combines with the big bubble? That's what happens. And then it dumps its genetic contents, okay, and coats them a little bit here. We have our reverse transcriptase. We're taking our RNA and we're converting it to DNA. That's what's happening here. Then we have an enzyme known as integrase, which is going to take that double-stranded DNA and it's going to put it within the host cell. The difference here in a provirus versus a prophage is instead of going through induction, where it will cut itself out, go into a virulent cycle, it stays with you forever. That's the biology of HIV. That's what makes HIV scary. That's what makes hepatitis scary. Hepatitis is another retrovirus. It stays with you and replicates with you, and it'll continue to produce those viral proteins. So recall, your eukaryotic viruses aren't going to kill the host cell. It's kind of like lysogeny. We can have infection occur for very long periods of time, even a lifetime. Now, depending on whether or not the host will exhibit signs and symptoms is going to be how we differentiate between a latent infection and a chronic infection. So a latent infection means that you're going to have initial signs and symptoms. The patient will exhibit initial signs and symptoms. Then it will go dormant. And dormancy can occur outside of the host cell. It doesn't have to be a provirus in order to be latent. Sometimes you just have that genetic material from the virus hanging out in that cell, doing nothing, until something changes later on. And then it turns those genes on again, shows signs and symptoms. This can be periods of months, it can be weeks, it can be years. Good example, aside from herpes, because herpes is up there. Can you want to think of another latent virus?
Shingles.
Shingles, what's it called first?
Chickenpox.
Chickenpox, right? Chickenpox goes systemic, affects all of your skin. Then after that, when it goes latent, it goes and hides in your nervous system, the ganglion cells, and then it'll manifest later as painful spores known as shingles. What are some other ones? Adenovirus, HPV,
Tuberculosis.
Epstein-Barr virus, so mononucleosis. Yeah, there's a few of them out there. Okay, chronic infections. It's where the host gets overwhelmed, basically. They can't get rid of it, and they're constantly deteriorating. It can lead to a depressed immune system much of the time. Right? So that's how HIV can become chronic. So HIV is both latent and chronic. We'll talk more about the biology of HIV later on with antivirals, but essentially what's happening is it's infecting your immune cells, and it's destroying these immune cells, it's evading other immune cells, it's changing the way that they work. And as it builds up in all of your immune cells, what happens is your immune system's depressed. It doesn't work very well anymore, and so you start constantly exhibiting signs and symptoms, which leads to a chronic infection. And that's when it becomes AIDS. Right, that was pretty much animals. There's only one slide that I have on plants. Okay, they're more similar to eukaryotic, obviously, than prokaryotic. The only thing that I really want you to know, underlined, positive single-stranded RNA genomes. What you pretty much need to know from this slide. Most every plant virus we find, and we don't really know why, always have this same archetype. It's also the way that they're transmitted is a bit different because plants don't get up and walk around, so technically they're going to use a lot more things like biological or mechanical vectors. Sometimes they rely on environmental vectors like water or wind. Let's talk about doing some lab work with them. So late 19th century, 1890s, we cared a lot about our tobacco. Everyone smoked back then. Doctors used to recommend it. They were trying to figure out what was killing a lot of their tobacco plants. Their plants were built, become modeled, and so scientists took it on because this was lucrative to figure out what was causing this. Dmitry Ivanovsky, Russian scientist, utilized a specialized porcelain membrane developed by Louis Pasteur that had 0.1 micrometer pores, and he would filter certain solutions after he would grind up these tobacco plants through it and try to isolate, take that filtered solution, reapply it to the plants. Why was he using this size, 0.1 microns?
Because they're 20 to 200.
How big is a bacterial cell on average?
200 micro.
About 1 micrometer. Right, so if you have pore size of that size, can bacteria get through it? No, right? It has to be something smaller. In general, there are smaller bacteria, like mycoplasms, but in general, most bacteria are going to get filtered out. And so he took that filtrate and he put it on the plant, and it started showing disease signs again. And so what did they think? They're like, hey, this is not a bacteria. We know the bacteria are about this size. This is something that other than a bacteria. So they postulated, they thought it was maybe spores, or they said, okay, there's maybe a new category of super small bacteria that we don't know about. Other ones, here we go, that's kind of what it's showing. Other ones said, no, I think this is some type of poison. Virus in Latin means poison. That's how we got the name for this, is they said it was a biological poison, what we now think of as toxins. Hey, but it actually wasn't a toxin, it was a virus, because viruses are 100 nanometers smaller, around 200. So it was getting through that filter here. And that's what it was showing in SEM. These are how small those pore hole sizes are, 0.1 microns. Then you have your bacteria that can't get through them. So that's how we initially started filtering them. He never really discovered that it was a virus because that was only found until like the 1930s when we had electron microscopy. But he still gets recognized as being the one who coined the term virus for viruses. So when we're going to grow these in the lab, we can do them either within an animal, a living organism, so we're going to call this in vivo culturing, or outside of an animal, which is in vitro culturing. So if you're growing this within a lab rat, right, in vivo, if you're going to take some tissue from that outside and grow it, it's in vitro. We can have both bacterial and animal cell cultures respectively for bacteriophages and eukaryotic viruses. In this case, what this is showing here, this is showing culturing of bacteria. These little spots you see aren't viruses that are necessarily growing. It is bacteria that they're killing. It's called a plaque. So y'all have done some inoculation on your plates in lab, right? Did y'all have to make what they call a bacterial lawn yet? You'll have to do that for your E-tests and your antibiotic Kirby-Bauer assays. So you basically take bacteria and you cover the whole plate to where it's like a film. Right? And so all of this beige here, they film, or what we call a confluent lawn of bacteria. Then they inoculate it with bacteriophages. And as the bacteriophages kill those colonies or those progenitors of those colonies, then you get plaques. So these are halos of non-growth, showing that those cells were killed by a bacteriophage. So yeah, bacterial lawn. Now, we do most of our virology studies with bacteriophages because of the fact that they're easy to work with and we don't mind killing bacteria. And this helps us understand how viruses replicate and grow over time. So we're going to look at 4 stages of viral growth: inoculation, incubation, burst, and burst size. And this is what it would look like if we graph this out. On the y-axis, we have the number of virions, number of viral particles. X is always time. So we start out, you're going to inoculate that sample, step 1, with a small amount of viruses. And if we were going to look for viruses in that media, we're going to detect a small amount. But then they all go away during the eclipse phase. So why do you not detect virions during the eclipse phase if you're going to sample that media? They're all inside the bacteria, right? They're hiding within the bacteria. Good. So they're eclipsed. They go through their cycle. 1, 2, 3, 4, 5. Boom. They all release at once. This is different than what we'll see in chapter 9 with the exponential phase of bacterial growth. This goes from none at all to all at once, because they all lyse and burst at once. So you go from 0 to many, and this is known as a viral titer or a burst size. So all of these that released will give you the amount of viral particles that are created. Pay close attention to that eclipse phase. So why are these types of studies important? For diagnosing viruses, yeah, that's important. Making vaccines, it's important too. And then et al. Is what number 3 is. Basically everything else, research studies. So let's make a influenza vaccine. As mentioned earlier, eukaryotic viruses have a tissue tropism. Okay, and it's kind of no different when we are reproducing these in vivo. And most of our flu vaccines that we make for all the people are done in chicken and turkey embryos. And that's kind of what's happening here. And they have to inoculate specific parts of that embryo, depending on the type of vaccine that you're making, in order for it to survive. If it's inoculated in that wrong part, then you would end up killing that embryo, and you're not going to produce enough of those viral particles so that we can attenuate them for a virus. Who in here gets the flu vaccine? You do? Good. Who? Okay, wait, I didn't do that because I screwed up there. Okay. Flu vaccine. How effective is the flu vaccine? That's good, isn't it? About 50 to 60% works. It's because we're trying to usually bioinformatically predict what strains will be occurring that year. A lot of times we can be wrong. We'll talk some more about that later in the course. All right, so let's do some in vitro growth. We're still growing in cells, but it's not going to be in the actual whole organism. So what we've done prior, and what we still kind of do, is you can take some cells, say you were trying to study a respiratory virus, you take some lung tissue and you culture those cells into a primary culture. What happens a lot of the times with animal cells when we're trying to do this in vitro is they'll exhibit what's known as contact inhibition and contact dependence. So they have to have some type of plastic or glass in order for those cells to stick or adhere on. And then as they grow to a dense culture, they stop growing once there's too many people. Right? You can think of this as like the subway's full, you can't fit another person on it. And so when it gets to that point, scientists have to pour some off, add some more media, grow it to a secondary culture. And typically after you get past a secondary or tertiary culture, it doesn't work. So it's very time, labor, and cost expensive. It takes a lot of time, it doesn't work. But we found a loophole to do this instead. What we use is immortal cell lines or transformed cells. So in this case, we're going to take some tissue that doesn't exhibit contact dependence or adherence dependence, or it doesn't exhibit contact inhibition. So they're going to just continuously grow. They don't need something necessarily to grow on, and nothing stops them. So yeah, where do you think they get that type of culture?
Cancer.
Sure. We take cancer cells. Think about what cancer cells do. They replicate unchecked and they lead to tumors. They take that and then we can actually do tests on these types of cells. And it's much better. And we use these today in a lot of virology studies. The main one we use are known as HeLa cells. They came from a young woman, a long time ago, back in the 50s, known as Henrietta Lacks. She was a 31-year-old woman from Baltimore who had cervical cancer. They took the sample without her permission. And this is, a cool little thing about the movie or the book if you've ever seen or read this. We still use them today, right? This is kind of like the gold standard of using an immortal cell line. But they took these from her without permission, and she died after that. Okay, and nobody really knew that they took it, but we use these and we've developed a lot of advances in virology studies from this. So what's wrong with this? It's unethical, right? There's ethical implications from this. So should we stop using HeLa cells? This this is an ethical debate. I'm not saying one thing's right or wrong. I want to hear y'all's opinions. Should we stop using HeLa cells?
No.
You think good outweighs the bad? Okay.
Did her family sue?
In the process.
Currently right now?
Last that I looked, they were supposed to be compensated. I think they got paid a pretty penny for it. It was only later years till the family found out. At first, they gave them copyright to her genetic material because they sequenced her genome, and they said, okay, you have control over her genome. And they're kind of like, where's that? Right? Y'all made a lot of money on this. So yeah, we're still going to continue to use this because this is a baseline standard for a lot of molecular work that we do. We can't just not use it anymore. That'd be like all of a sudden iPhone gets canceled and everyone's like, well, no longer do these products work. That kind of stuff, right? Question?
Why don't we get like new cancer cells from like an existing person that has given their permission?
We still do, but they are different, right? You have all these protocols, standards, they're produced, mass produced, sent out. There's a lot of research that has been done with them. And so if you are doing research with this, that's like all of a sudden being given like, oh, you think these are the same, but they're not really the same, right?
I mean, this is like, they already took it, they might as well keep using it.
Right? Yeah, here's my thing, compensate the family. What are y'all whining about, right? It does help people, and I think the family would be happy to go, yes, we want to continue to help people, but use this as an example, teach about it so unethical stuff like this does not happen again, and pay me my money. So I think that's what's happening. Really good movie, really good book. You need something to do on Friday night, you're done studying, there you go. All right, so let's detect viruses in people. Now we're getting to diagnostics. We can look at just by the physiological change on the cell, so the effect on the cell that we're going to call cytopathic effects. We'll do it by an HAI assay, or a hemagglutination inhibition assay. We'll do it through some PCR, and then we'll do it through an ELISA. So CPEs, basically everything that you know about how a eukaryotic cell should look, or even a prokaryotic cell in some cases, it's going to affect it. It's going to cause it to change shape, die, multiply its nuclei, shrink its nuclei, form new types of organelles or vacuoles. It's just going to make it look really weird. Other major things that you can see are the formation of tumors, or the effect affecting its host genome. So this is just some examples of cytopathic effects. You don't need to know the pictures, but you should know this list here. But yeah, you're looking at stuff under a microscope. Practice. Look at it. You want to know what's normal versus what's not normal. You can save somebody's life. Hemagglutination inhibition assay. So as we get infected with viruses, what does our body do in response to fight that? What? Okay, elaborate. Yeah, so say you get sick with the virus, how does your body fight it?
It starts to go hot. Starts to
You
raise
get
the heat.
hot. Okay, yeah, so you go through normal types of acute inflammation. What else? Why do we get vaccinated?
So our body recognizes it.
Okay, when you're building immunity, what are you producing? Antibodies. So we're going to use the fact that if somebody is sick with a virus or has had a virus, that they have antibodies within their blood to determine whether or not they have a specific virus. So this is known as an HAI. So what we do is we take a patient's serum, okay, not to be confused with plasma. Serum does not include the clotting factors in the blood. Okay, so if you're taking the serum, you're going to have your antibodies within there. They're going to combine a known virus with a patient's serum, okay, in this first step. Combining a known virus with a patient's serum. If the patient has antibodies, what that means is the antibodies will bind to that virus. If the patient hasn't been exposed to the virus or doesn't have it, they will not have antibodies. Next step, they add in red blood cells or erythrocytes. Viruses have those hemaagglutinin spikes. Remember the H spikes? That means they agglutinate blood. If the virus can bind to the red blood cells, it's going to cause it to clump up. So, let's work through whether or not this clumping is inhibited or occurs. Then B here, you took the virus and the patient's serum. They don't have antibodies. Antibodies don't bind the virus. So instead, the virus binds the red blood cells, causes clumping. Here, you take the virus, you add it to the patient's serum. You do have antibodies. These bind to each other, so the virus can't bind to the red blood. The virus can't bind to the red blood cells. No agglutination. Who has the virus? Person B or person C?
Person B.
C. Why does C have the virus? Because they have the antibodies for the virus. Does everybody kind of see how that works? Okay. Agglutination inhibition is indication of viral infection. HAI assay, hemagglutinin inhibition assay. Let's work through it again. You start by getting a known virus and you add it to that patient's serum. If you have been exposed or you currently have the virus, you should have antibodies for it. So the virus and the antibodies are bound up in step one. Then you add red blood cells. Red blood cells can't interact with the virus, so you have no clumping. You see no clumping? That is indicative of having the virus because they have the antibodies for it, or that means they've had it before. And antibodies can stay in your blood for weeks, months, years. So all of this does is tell you that you may have the virus now or you've had it in the past. But if you're exhibiting signs and symptoms, then you can go, okay, well, there you go. Right, you have it currently. Are we clear? If not, then I can go over it at the end of this lecture again for those of you that want more. A nucleic acid amplification test, or PCR. Basically, we take a small amount of genetic material, we design some primers that are unique to that gene or genetic material for that specimen we want to identify. So we want to know whether or not you had COVID. You're going to take that small amount of material and you're going to make it into a lot. The way that this happens is through successive cycling of attaching something that is known as a primer to that DNA, splitting it apart, replicating, splitting it apart, replicating, splitting it apart, replicating, go through like 30 or 40 cycles of this. You can go from a tiny amount of genetic material to a lot. And this is what they use in your NCIS and stuff when they find a little bit of blood or semen or whatever it is. And they're like, okay, I can amplify this small amount to a lot and use it for a lot of testing studies. If you wanted to go from RNA to DNA, then you use reverse transcriptase PCR, right? Polymerase chain reaction. You would just take that RNA, convert it to DNA, and then replicate it through polymerase chain reaction. So what next steps that you can do with this, I've done this thousands of times, you can take that genetic material, so you amplified a gene, and that gene is 200 base pairs long, and you can go through gel electrophoresis. I might have done some of this in high school. Anyone do a gel? Well, what we do in this case is you take basically a jello block that you make, and it has these vertical lanes. You can see N lane, 1 lane, 2 lane, 3 lane. I'm not going to get specific on these questions. I'm just teaching you. You have a marker lane. This is a comparison lane of sizes of genetic material that we know. And then you put your amplified DNA in each of these wells. You put that jello block into an electrolyte solution, and then you hit it with some electricity. And since DNA is inherently negatively charged, it's going to flow to the positive anode. So as DNA goes from negative to positive, big DNA moves slower through the jello, small DNA moves faster and further. So you get further down here, these bands are only 100 base pairs, whereas these would be like 500. So we can confirm whether or not somebody has something like COVID by amplifying a gene, setting it to a gel electrophoresis, and be like, if we look for a band at size 100, it is confirmed. If it's not there, then they don't have it. Really fun to do, very hard to do. I've done it 1,000 times. So what you expect maybe is something like this on the left. You're lucky if it looks like that, but 90% of the time it's going to look like this. And you're like, wow, I just wasted 8 hours of my life. Time to do it again tomorrow. That's what grad school is a lot of the time. Then last thing here for detection are the enzyme immunoassays, so an ELISA. Enzyme-linked immunoassay, immunosorbent assay. It's also called a sandwich ELISA sometimes. All you need to know for this, I'll explain it, but all you need to know for the question, this is a way to detect viruses directly or indirectly, right, through antigens and antibodies. Detection of viruses directly or indirectly through antigens and antibodies. With PCR or nucleic acid amplification test, you're looking at genetics. Here, you're looking at antigens and antibodies. So this is showing a direct assay, not an indirect assay. On the left-hand side here, we're going to have a positive result. On the right-hand side, we won't. You're actually going to do this in lab on one of the last weeks when you're going to test for COVID. So what we have is we have these wells, or these membranes, and on these membranes, they're coated with a specific antibody. So these little V's here are little antibodies that will detect an antigen, the little diamonds here. So as you have these wells that can detect them, you add the patient's sample. If they have it, then the antigen will bind to it. Okay, you do some washing and some cleaning. Then you're going to add another reagent. This is going to be a conjugated antibody that will also bind to that antigen. What a conjugated antibody is, if we go back to immunofluorescence, is an antibody that has some type of enzyme that will change colors whenever you add an ingredient. So we add our conjugated antibody. Yes, it's going to stick on. No, since we don't have the antigen, it washes away in the next step. So when we get to the last step and we add that reagent that's going to cause that enzymatic reaction, usually a chromogenic change, it's going to turn a color. It's going to say, yes, pop. No, not positive. Has anyone in here ever done this? You sure? Someone here's taking a pregnancy test. No, don't answer that. Someone here's taking a COVID test where you look for 1 line versus 2 lines. There you go, you did an ELISA without knowing it. Think about it, you took those samples, you added it to a membrane that had an enzymatic reaction. If it changes the color or a color shows up, that means it was bound. If not, right? They can get a little bit more complicated than that, and as you'll see in lab. Okay. All right. Now this will go really fast. Just talk about these other ones: viroids, virusoids, and prions. Okay, not viruses, but viral-like. Viroids, not a virus, okay, but it's similar to it. They're usually much smaller. Here's the difference: small circular RNA that can self-replicate. We haven't really found any viroids that affect human health necessarily directly. A lot of these ones are just going to cause agricultural problems. The example here, potato tuber spindle disease. It just makes your potatoes look bad. I'm bringing up potatoes again because I love potatoes. But there's a lot of viroids that affect important crops: avocados, tomatoes, peaches, a lot of your stone fruits. So we do have to worry about these to a degree because we don't want to knock out our food chain and food supply. I do like going to the grocery store and having a lot of good produce. They just opened up a Sprouts by my house, and that's the first time I've ever been there. It's fantastic. Okay, so it can self-replicate. Doesn't have a protein coating. That is why it's different from a typical virus. Remember, virus has to have protein, genetic material. This is just genetic material. Virusoids, even smaller, maybe only a couple hundred base pairs long. How it is different than a viroid? It does not self-replicate. It has to bring a friend. It has to bring what's known as a helper virus. So it has to have a little friend virus that goes with it and replicates it for it. What it usually does in the cases when it causes diseases and stuff is just amplify that disease. So if that virus is causing a disease and a virusoid comes along, it usually brings extra genes that make it worse. And they're trying to show that there's a simpler satellite virus found within hepatitis, known as hepatitis D, that makes hepatitis worse. And lastly, prions. This blew our mind not too long ago, back in the 80s. Before, we thought all infectious agents had to have genetic material, even if they didn't have a protein capsid. But no, these are just proteins. This is just a rogue protein that is misfolded. It doesn't have the correct tertiary or quaternary structure. Misfolded that causes other proteins to misfold. Untreatable, very hard to kill, very hard to get rid of. This will kill you, no matter what. It is contagious too. Y'all heard of mad cow disease? Hopefully. Mad cow disease comes within cows. It's similar to like wasting disease in elks. It's similar to scrapies in sheep. Basically what it's going to do is attack the nervous system and cause it to perforate and have holes in it. And yeah, your brain, like Swiss cheese, isn't going to work. Whenever this gets transmitted to humans through eating something like contaminated meat, Okay, it causes what's known as Creutzfeldt-Jacobs disease. Creutzfeldt-Jacobs disease. So that's a form of transmissible spongiform encephalopathy. So TSE, transmissible spongiform encephalopathy, is caused by prions. That's what it makes your brain look like. Normal brain, yeah, nice and full. This one, full of holes. Get it not only from eating animals, but this can be passed down hereditarily. In the case of CJD, that happens. It can come from things like organ transplants. Really difficult to destroy. You have to incinerate it at very high temperatures. A lot of chemicals aren't going to affect this. Okay, there's no type of antibiotics for this. If someone has a prion disease, we have to be careful, especially with the person who's deceased, on how we get rid of them. Now, you're not going to get, even though it is contagious, it has to be through like blood, saliva, some type of other mucus. You're not going to get it from hugging the person or shaking their hand. Be cautious, be aseptic, but it's not spread casually. It's only spread through those other means. So here's a few examples of this. You don't need to know this table, just know that it can be spread through heredity. Always one that I do like to point out is Kuru, through cannibalism. That's a pretty cool thing, right? No, not really. That we do study a lot of cultures. I used to be an anthropologist, where you study the spread of disease through cannibalism. How do we feel?